The Institute was renowned for its work in immunology, and we convinced Nossel that the time was ripe to embrace molecular biology. We wanted to tackle the two great mysteries of the field at that time. And Paul has referred to this already. How does the body generate the billions of antibodies it uses to fight infections? And how does a single antibody-producing cell switch from making one class of antibody to another? Again, the first years were a struggle until a technological advance, and this was, of course, the advent of recombinant DNA technology pioneered by Boyer and Cohen in California. Labs around the world raced, including ours, raced to set up this technology and unlock the secrets of antibody genetics. Suzumu Tonegawa, then at the Basel Institute of Immunology, got there first. To everyone's total amazement at the time, he found that the variable region of these antibody molecules, the parts that are really critical for capturing foreign proteins such as virus molecules, are not encoded in the germline as complete sequences. Instead, they're fragmented, and each lymphocyte has to actively assemble a complete sequence from the individual elements selected at random. And because so many different combinations are possible, the total number of antibody specificities that can be generated becomes billions. So this was a very exciting time, and we managed to make some significant discoveries. I think the two that we're most known for is that antibody class switching involves another remarkable DNA cut and paste job, and we also helped to enumerate the number of antibody gene elements in the germline. These years were also productive in other directions. Jerry and I had two wonderful daughters, Karen and Carly, who opened up wonderful new horizons for us, and we count them as our very best experiment. But back to the lab. In 1975 came another big event in science. Jerry and I were electrified to learn that Harold Varmus and Mike Bishop had found that every mammalian cell contains genes that are very similar to those that are found in acute leukemia viruses in cats and rodents. The implication of this stunning finding, which was to earn them the Nobel Prize, is that each one of us actually harbours the seeds of cancer, genes that can turn rogue if they're mutated or expressed in the wrong cell type or at the wrong time. We began to read voraciously in this new field, very keen to become part of it. Now, we'd been using Burkitt's lymphomas, which are tumours of antibody-producing cells, as tools for our immunogenetic studies. And we found out that Burkitt's lymphomas have a very particular genetic abnormality, which results from two chromosomes being broken and rejoined to the wrong partners. Now, we knew, as I've just told you, that antibody gene assembly involves cutting a chromosome at several points, throwing away certain bits, and then rejoining the ends. So we wondered to ourselves whether the chromosome translocation that I've just described was an aberrant antibody gene rearrangement. Could the broken end of an antibody gene locus have been accidentally linked to a cancer-provoking gene on another chromosome? And so we set out to test this hypothesis using an antibody gene as a tag to clone this putative oncogene. And sometimes even your scientific dreams come true. Sure enough, linked to the antibody gene in every tumour we analysed, we found a gene called MYC, the mammalian counterpart of a cancer-provoking gene that had already previously been identified in an avian retrovirus. As is so often the case in science, 3 other labs independently reached this exciting conclusion at the same time. It seemed almost certain that we'd found the root cause of Burkitt's lymphoma. But at this stage, it was guilt by association only. We wanted direct proof. And to get that proof, we had to turn to yet another technology that was just being pioneered at the time in the US. Richard Palmiter and Ralph Brinster had learned how to genetically manipulate mice by introducing DNA into fertilised mouse eggs. We sent Palmiter and Brinster our MYC gene, linked to the antibody gene control elements. I'll never forget the day when they rang up from Philadelphia and told us that 100% of the mice carrying that additional modified MYC gene were coming down with lymphomas. We had found the definitive proof that we were looking for. Our MYC mouse has become a very powerful model that's still used around the world for studying cancer development, and the MYC translocation became a paradigm for isolating other oncogenes from different kinds of lymphomas and leukemias. Our own lab went on to study several other oncogenes activated by chromosome translocation, but one in particular stands out because it totally shifted our research interests yet again. This is a gene called BCL2, which US researchers had cloned from a translocation that was associated with follicular lymphoma, a different kind of lymphoma. But the function of this protein, the protein encoded by this gene, remained a mystery until 1988, when David Vo discovered during his PhD studies in our lab that, as Paul has told you earlier tonight, BCL2 promotes cell survival, not cell multiplication, cell survival. It enables cells to survive adverse growth conditions. Of course, using the model we'd already established, we again created a different kind of transgenic mice, BCL2 transgenic mice, and we went on to show with Andreas Strasser that overexpression of BCL2 is indeed oncogenic, especially when combined with another type of oncogene, such as the MYC gene. This was really a major milestone in cancer research, because for the first time it was realized that cancer development involves not only accidents, genetic accidents that promote cell division, but also those that block apoptosis, this physiological process of cell death. We and others found out after that BCL2 has many relatives, and while some of these also promote cell survival by blocking cell death, others instead promote cell death. And together, this family of proteins constitutes a critical switch that determines whether at any moment a cell should live or die in response to the complex signals it's receiving both from within and from without. So the BCL2 family is now our central focus, and we formed a consortium with several other labs at our institute to decipher the molecular circuitry of cell death. And we hope to use this knowledge to develop much more effective drugs to use against cancer cells, and indeed the prospects are looking good. So there, in a nutshell, you have my scientific journey. It seems like it moved from one totally different field to another, but as I hope you've seen, there's a thread of connectedness to all of this. I feel incredibly fortunate to have been part of such a revolutionary era in biology, but the best is still to come. How I envy young scientists starting out their careers now, in this age of the genome revolution. What wonderful prospects lie ahead to fight disease and ill health, to conserve our planet and to promote economic prosperity. And I hope that in the future that lies ahead, at least 50% of those driving science will be women. It concerns me greatly that too many women continue to opt out, discouraged by the difficulties of combining career and family. We simply cannot afford this brain drain. That's why I think the Pearl Meister Greengard Prize is so significant. In a sometimes hostile world, it stands as a rare beacon to inspire and encourage women of science. On behalf of all of us, thank you, Dr. Greengard. Thank you, Ursula, for your generosity and vision. Thank you for your attention.